different way. So there's a lot happening on this slide, but I just want to orient you. So here we have an embryonic stem cell. As soon as that stem cell divides and its daughters start taking on a fate, meaning they are no longer pure stem cells, but they might be committed. So committed to a lineage, meaning they're starting to turn on genes that are specific to one type of cell lineage. And then once that is further along in the developmental time scale, then that would be a differentiated cell. So this is kind of a measure of time and of level of development and differentiation, and also a loss of potency. So this would be the most potent, pluripotent. This might be multipotent, and this would not have a potency. It would be committed to a specific kind of cell. So then in that context, if you think about just 5 random genes, but these genes have been studied, and this first one shown here is a pluripotency gene. So that gene, when it is expressed, conveys onto that cell pluripotency. Some of these other genes might just be developmental genes that are involved in certain cell types, whether it's forming mesoderm or ectoderm or some specific cell properties. And then the last gene in this cluster might be the late differentiation gene, so something highly specialized, whether it's for a gut cell that has to secrete some sort of an enzyme for digestion, something like that, that would be very far in the developmental spectrum. So when you look at an embryonic stem cell, you've only got this pluripotency gene on, and all these other genes are turned off. And as these cells start committing and going into more differentiated state, this pluripotency gene is now off, and other genes are starting to be turned on. And as it gets to the most differentiated state, you might have a few genes on